What's up to you all pre-matriculants? Welcome to it. This is your favorite show where you learn more and learn extra. My name is Abram and today I'm with Aslam. It's the A Squared team in the building. Aslam, how are you?
Do you realize what you just said? Welcome who?
Pre-matriculants.
Pre-matriculants.
Yes.
Oh, I didn't hear the pre. I was worried.
It's fine, I understand. Yeah, the years have gone by.
The years have gone by.
It's almost the end of
My,
the year.
my tympanic membrane must be wearing off.
Maybe. You're not the only one. John has that problem too. Are you good though?
I'm fine.
Right. What are we doing today?
Today, for the grade 11s, we are simply going over questions in paper 1. That's all.
That's all we're doing.
Yeah. They're writing exams. Exam season is in. Next week, most of the provinces, they start their final exams internally as well. So most of these learners will be involved in exams. So therefore, we are pushing with revision paper 1 this week, and next week we do paper 2.
Nice one. And guys, relax, take it easy, make sure that you focus. And tell you about focusing, this show is proudly sponsored by MTN SA Foundation. So thank you so much to MTN SA Foundation for making this show possible for all of you guys. And thank you for tuning in. To help you furthermore, we've got notes for you guys. Go to our Facebook page. It's facebook.com/learnextra. But in case you also want other subjects and you also want other notes, go to learn.mindset.co.za. Follow us on Twitter at LearnExtra. During the show, I'll be telling you more about the giveaways that we have. But for now, learning.
Double A.
Yes, double A.
Double A, yeah?
Yes.
You didn't say that part. Welcome, grade 11s, in this unusual slot with this unusual face, or these unusual faces. You're not used to our faces, both of us, and you're not used to this particular slot. The slot is normally the grade 12 slot, 6 to 7.
It's a promotion already, Aslam.
It's a promotion already for them. There you go. You see, Abram will always find the positive thing about everything. That's great. That's great, Abram. Well, let's go straight into the challenge question. Mindsetters. The question is very straightforward. However, you need to think carefully about it. It says, explain how the functioning of the kidney will be affected if the ureter is blocked by a large kidney stone. Hint, worry about that fellow, the ureter first, and think about the functioning of the kidney, and put those two together, and you should be on the right path. And that you should do with any question that asks you about a part that is damaged, etc., etc. Always look at the part, what is its role in the whole scheme of things, and you should get around it. Second one, explain how the body responds to dehydration on a very hot day, or when one is involved in strenuous exercise. So the 3 things to worry about here: dehydration, hot day, or strenuous exercise. These are the things you need to look at. So the first one is about kidney stones, and the second one is about dehydration on a hot day. You put your answers on the page first. Let's go straight into the questions. This one here, question 1, adapted from paper 1, standard grade native paper, prep paper 2010, sorry. The apparatus below was set up by a learner. She made sure that the test tube was initially full of water. Okay? Reading the instructions there, looking at the diagram. There's a diagram. It's telling you that there's a water plant there. There's a funnel that is inverted. There's a beaker that has water, all of this here. And there are bubbles of gas that are noticed inside the test tube that is inverted as well on top of the funnel. Remember, all of this equipment is underwater. And here we have some blocks, rubber blocks or other blocks, to lift the funnel up a little bit. Good. So first question there is, what is the aim of this investigation? Firstly, guys, let's get used to some terminology. Aim means, why are we doing this investigation? What do we intend proving, illustrating, finding out, etc.? Why are we doing this? Okay, so that's the first thing. And the aim of this particular investigation, you would know we're using a plant here, and in your syllabus, the plants with a little bit of gas should tell you that this comes from that packet of knowledge in the KEPS document that is called photosynthesis. So obviously this experiment has to do with photosynthesis. Why am I going to that length of explaining that? You need to contextualize all questions. In your mind, you must be able to open the right file. Because if you're saying that this got to do with photosynthesis, then the right file is going to open, and from there, the rest of the work can help you. So the aim of this particular investigation then is to show that oxygen is released during photosynthesis. And you can already see the clues. There's the plant there, and there's the gas there. We're assuming that this is oxygen, the bubbles of gas there. Why was the test tube filled with water? Why did we fill this test tube with water? Here's your answer. One of the part of the answer is the fact that you're using a water plant, and we don't want the gases to escape, etc. To make the apparatus airtight, want to find out whether any gas is produced, therefore must not have any gas to start with. So what do we mean by that? If this beaker and the test tube itself here, this test tube in this area here, if it had air inside already, we are not going to be sure that the gases that we are seeing are oxygen, or came from, rather not that they are oxygen, that they came from the plant. And they're not there already. So what we're doing with the test tube, when we're filling this test tube with water, we're pushing any of the gases that are there out. So the air is being forced out from the test tube itself. Why was it necessary to leave the apparatus near a warm, sunny window? Now, if we're talking that we are talking about photosynthesis, this should be easy. Warm, sunny window. Sunny, giving you light, and warm, giving you the right temperature for photosynthesis to proceed. So the idea is to allow photosynthesis to play. Without light, photosynthesis cannot take place, and the enzymes in a plant are also sensitive to temperature. So the right temperature, generally about 25 degrees Celsius, is more or less a nice temperature for photosynthesis to proceed in plants. Name the gas that is present in the bubbles. I've already given you that, and that is oxygen. Describe one way in which you would test for the gas named in question 1.4. How do we test for this gas? Okay, the description is simple. We can use one of two ways. One is obviously we've got to remove the test tube from the funnel. That's the first thing for both ways. We can either insert a glowing splint, end of a wood splint, into the test tube, and if it glows brighter or bursts into a flame, then we say oxygen is present. Or another method is we can take steel wool or some other metal and throw it into the test tube. Generally, that steel wool would rust to prove that oxygen is present as well. Now, just by the way, a glowing end of a wood splint, that means we burn wood and we kill the flame. So we see that that part is still red. That's the glowing end. Just so that you know what we are talking about. That's the glowing end. When we put it into the test tube, it should glow brighter, showing that oxygen is there, and we'll talk about that just now, or it can burst into a flame. Why? Because oxygen supports combustion. This is what we are showing there, and that is why that would be a positive test for oxygen. There are other means as well. We're looking at these two that are prescribed. It's good enough for you. Good. Let's move on. Let's just go back one step. Now remember, they can even ask you to set a hypothesis for this particular investigation, and your hypothesis must involve what you're testing for. You want to see whether the gas released by the plant is oxygen, so your hypothesis will be around that. For example, you could say that the gas released by plants during photosynthesis is oxygen. Or you could say is carbon dioxide or methane or any gas that you choose, because that's your hypothesis. The experiment then would have to change to meet your particular need for your hypothesis. And If you said methane, for example, and you tested for methane and that was not true, then you would have to reject the hypothesis and start again. So you start with another hypothesis, you do the experiment, and you come to some conclusion as well. So worry about that as well. Also, you can ask you questions on the independent variable, the dependent variable. Make sure that you are aware of these terms. For example, the independent variable is the variable that we, the investigators, can change. The dependent variable is the variable that will be a result of our changes to that experiment, and we, the investigators, are not in control. Generally, in life sciences, it would be the rate, the percentages, the change in height, change in growth, etc., 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 would be the dependent. And time, pH, temperature, etc., because we can change those things, will be the independent variable. We'll talk about that later again. Next question, more on theory now. Adapted from the paper 1 standard grade later 2010. The diagram illustrates the internal structure of a leaf. Answer the questions based on it. So there they give you a lovely diagram of a dorsiventral leaf. And they've asked questions. The first question is identify A, B, and E, as well as cells C and F. So they're pointing out to you that these are particular cells that they want to know. These others are structures there. So it's A, B, and E. A, this is a region, you can see it's an area there. This would be the palisade mesophyll. B, this will be the upper epidermis. Even though this line is pointing to a particular cell, they didn't say, the fact that they're telling you cells here, you must worry about cells in the next part of the question, not in the first part of the question. Okay? And E, that's air spaces, that's an easy one. As well as C and F. So C now, this one here, what type of cell is that? That would be a parenchyma cell. Or we can say it's an epidermal cell. And cell F, these two of them, these are known as the guard cells. How we know that they are guard cells? In between the two, you'll see a space there, in that area there. That space is known as the stoma. Don't confuse the word stoma with stroma. The stroma is the fluid matrix in the chloroplast. Stoma is a space generally more on the lower epidermis, but can be found on the upper epidermis as well, to allow for gaseous exchange in the leaf. Okay, let's go to our answers there. The palisade tissues, the cuticle, the B, the cuticle. No, the line has moved, okay? In this particular case, sorry, the line has moved slightly there. This line is now pointing to the epidermis. I want you to look at that carefully. Look at where it is now. The cuticle is this line on the top there. That would be the cuticle. But these cells, this thing is pointing to the cell here, as you can see. That is the epidermis. So the line has moved slightly, unfortunately. Spaces, epidermal cells, guard cells, we got them all other than that right. State the role of each of the following for the process of photosynthesis. B. The cuticle there now, if it was the cuticle, obviously our answer is going to be different. It is transparent. Well, anyway, for that case, epidermis is also transparent to allow light to pass through. And in this case, because it was the cuticle, if it was originally, it prevents excessive loss of water. But if it was the epidermis, then this part will fall away. This answer, the second part of it, is for the cuticle. D. What was D again? D is pointing to those organelles inside the palisade mesophyll, and that would be the chloroplast, and that's obvious. It absorbs sunlight, or it traps sunlight. It contains chlorophyll for photosynthesis. In other words, the chloroplast organelle is the part that has the pigment called chlorophyll, which will trap the sunlight for photosynthesis. E, going back to the diagram, is the air spaces, obviously, to allow for gaseous exchange, etc., and water exchange. Tabulate two differences between cells C and F. Cells C are the epidermal cells, and cells F are the guard cells. What are the difference? First of all, the epidermal cell has no chloroplast. The guard cell has chloroplast. The epidermal cells are thin-walled. The guard cells have this structure like that. The outside is thin, and the inside is thick, so they are unevenly, notice that the guard cells are bean-shaped as well, unevenly thickened cell wall, and they are brick-shaped, flattened brick-shaped, and those ones are bean-shaped. Notice in a question like this, we also give you a mark for the table. You must put the table. So even if you don't know the answer, at least put the table, Abram. You get 1 mark for Mahala. Huh?
Wow.
Tabulate. It tells you tabulate, draw a table. And you can just write cell C and cell F there. Just remember that. So when you are looking at that particular question, you don't make a mistake of labeling incorrectly. With that, A, B.
Thank you so much, A. Well, guys, during this break, you do have the challenge questions, so send us through your answers. We'd like to see how you guys are doing with that and help one another on the page. If you don't know where our page is, it's facebook.com/learnextra. Stay tuned. Welcome back, awesome Mindsetters. Now, after saying that, I think my job is simple. Getting yourself involved or part of our competition, it's easy, guys. There are only 3 steps. All you need to do, one, is to buy yourself this official Safeways K53 Learners and Drivers Manual, or download the cool Android app available from Google Play Store. And secondly, like our Facebook page and like the Safeways K53 page, which is facebook.com/Safeways K53. And lastly, enter your details because we'd like to get your details. The entry form is also on our page. Check, there's a link there which is called Win a Chevy Spark L. You click there, you fill in your details, done, you're entered, and you could win a Chevy Spark L. So guys, you've got only 3 weeks left before we announce the winner, Aslam. How good is that?
I need that Chevy Spark.
You need that?
Yeah.
Unfortunately. Me and you.
We're out.
In case someone doesn't win, when you have bought the book or the app, make sure that you keep the receipt, because if you don't have the receipt, you're not going to win.
Yeah,
that's the trick. So over to you, Aslam.
Thank you, AB. Welcome back, Mindsetters. I hope you had a refreshing break. We move on to question 3. The diagram, notice now we've answered a few questions on photosynthesis. Remember, you also have to know the process of photosynthesis very briefly, what happens in the light phase, what happens in the dark phase. You must know that as well. So, although the questions didn't directly ask those questions, you need to know that as well. Okay, and there are other experiments that you also need to concentrate or focus on as well. We've taken one such experiment. And the idea is to question you on the skills of experiment. What are you expecting in an experiment question? So you need to know that. So you need to know the method you followed, etc., for those little experiments that you did. Good. Let's move on. Now, let's see where does this fit in. Already, it tells you, study the diagram and answer the questions that follow. And the first part of the diagram tells you part of the human gaseous exchange surface. So obviously, guys, that should click the icon in your mind that says gaseous exchange in humans. You open that file and you keep it ready to retrieve information from your database on this section. So the first question always, when I give you a diagram, 9 out of 10, the first question is going to be give labels. So let's go to the diagram. So we have to label A, B, and C respectively. This is A, B, and C there. So A there would be a blood vessel, as you can see coming. No, sorry, not a blood vessel. What are you talking, Aslam? Right, here, look at A. What's A now? A is part of a tubule that is breaking up into smaller tubules here, as you can see here, or the air sacs as we are seeing in this area. So if we look at the gaseous exchange, coming from the nasal passage comes the trachea, after the nasal passage, obviously, and the trachea branches into 2 bronchi, or 1 bronchus, many bronchi, and these bronchi branch again into bronchioles. And these bronchioles end with alveoli. So this part there then will have to be the bronchus. In this case, one bronchus. B is pointing to the sac here. That is the alveolus. One, alveoli, plural. And C, as you can see, look at what's happening there. There's an arrow going out and an arrow going in, and inside there's branches. Those will be the capillaries. So the bronchiolus, did I label it properly? Bronchiolus, sorry. I labeled that incorrectly, and I showed it to you here, and then I still made the mistake here. Watch how careless we can be. Remember, I said first is the bronchus, then the bronchioles, and then the alveolus. And this one is coming straight to the alveolus, so this one here will be the bronchiole. Okay, and the alveolus and the blood capillary or arteriole. Describe one major difference between the composition now, the composition of the blood in vessel D and vessel E. Go back to the diagram. Vessel D is around here in this area. And vessel E is in this area. So this is these are the air sacs. This one here is closer to where the blood is coming in. And this one here is closer to where the blood is going out of this area. So it's obvious that it's got to do with oxygen, because the blood comes here to get oxygenated. That's one of the most important differences, but let's see what we are saying. Blood D is deoxygenated, while blood E is oxygenated, as I told you, or blood D contains less oxygen or more carbon dioxide, while blood E contains more oxygen or less carbon dioxide. So it's different ways in which you can say that, but that's the whole idea is about oxygenation and deoxygenation. Name two features of the structure shown in the drawing which makes it efficient for gaseous exchange in humans. Now they're talking of the alveolus, obviously. For any gaseous exchange surface to be efficient, it must be large, it must be thin, it must be moist, it must be protected, it must either be in direct contact or well ventilated, and for large, moist, and it must have an efficient transport system. So we need to say how this particular one meets these requirements. In other words, if you are applying for a job, my job is that you must be my receptionist. I have a list of tasks that a receptionist must be able to do. And you must be able to meet those requirements to be the successful candidate. In the same way in the body, all these different parts in our body, they have a job to do. And when we talk about how are they efficient or how are they suitable, we are saying what do they have in their structure to make them the lucky winner to do that particular job. Not lucky, but the correct chosen one. And there we have it. So first of all, there's many alveoli, and this increases the surface area. This one said name, remember that. We're just giving you the suitability. But if it says describe or explain the suitability, then you're going to say it has numerous alveoli to provide a large surface area. There is presence of blood capillaries. In an explained question, you would say for transport. It has thin walls, obviously, for easy diffusion. It has a moist surface to allow oxygen to dissolve, because oxygen can only be used in our body after it's dissolved in moisture. Okay, we could also add that large, thin we've covered, moist we've covered, transport system we've covered. We can also add that because it is inside the lungs, it is protected. It's found inside the lungs. So the lung itself protects it, and the lung itself is protected by the rib cage, so you could add that. But these are the more important ones there. Describe the mechanism of exhalation. Exhalation means how do we breathe out? And that's fairly simple. We start off with the diaphragm. The diaphragm relaxes and becomes dome-shaped or convex. So in other words, this is what's happening to the diaphragm. Now, the minute that's happening in the chest cavity, if the diaphragm is becoming dome-shaped or convex, you can see that the space here has decreased. The volume has decreased. Okay? The external intercostal muscles, that means outside of the rib cage, they relax. And the internal intercostal, inside of the rib cage, they contract. So if they're going to contract, they're going to pull the rib cage back to its normal position. So in other words, so from top to bottom, we've decreased the volume. And from side to side, we've also decreased the volume. And what does this do? The total, we'll worry about this one just now. The total volume of the thoracic cavity decreases, and the pressure on the lungs increase. That means on the lung compared to the atmospheric pressure outside. So the lungs now have a higher atmosphere, have a higher pressure compared to the outside. So air rich in CO2 is forced out of the lungs. Okay? Now your problem is you need to know inhalation and exhalation. And that's where the problem is. While we're learning it now, it's easy because there's no stress. But get into the exam room. When they're asking you exhalation, you're scratching your head and say, ish, what must contract, the diaphragm or must it relax? Right? It's difficult, Abby.
It is.
But now, I'm saying to you, learn only one.
Okay.
Must be a crazy teacher, this.
Yeah.
He's
what
not
I'm
asking you
trying
to learn both. He's only asking you to learn one.
You are weird.
I am definitely weird. Why must you learn one? Because the other one is the opposite. Let's do that very quickly. So if you want to know inhalation now, during inhalation, the diaphragm will contract and becomes flat. In other words, from that position, the diaphragm goes that way. The intercostals will relax, and the externals will, where's the external? Will contract. Look at what I'm doing, AB. I'm only changing one word at a time in that sentence. So the rib cage moves apart, or is lifted, we can say. What has happened because of this? The volume increases. And if volume increases, then pressure on the lung decreases. To equalize this pressure, what will happen? Air rich in oxygen is inhaled. Now, what I'm saying to you, mindset is very simple. Learn the one, the other one is the opposite. So if they ask you inhalation in exam, and inhalation is what you know, sharp. If you know inhalation, but they ask you exhalation, AB, still sharp. Because it is the opposite to what you know. So in that way, you know for sure what you know, and you move from there. By the way, here, the abdominal muscles contract during vigorous breathing. In other words, when we have to breathe in heavily, then the abdominal muscles also help to take in air and release air at a faster rate. But the more important points are all others besides that point there. Now for an application question. The graph below represents the result of an investigation using a spirometer. A spirometer is an instrument used to measure the amount of air that enters and leaves the lungs during breathing. They're telling you, they're not expecting you to know what a spirometer is. They're telling you what it is. And they have given you a diagram there, a graph, to show you what is happening. The line going up is breathing in, the line going out is breathing out. So here you see something normal, all of a sudden, ooh. In other words, it was like that, and then all of a sudden, AB, at this stage, what's happening? You're breathing heavier in this area. Okay? So there, they've given you that. You needed to read. Look at the graph again. It has time, which is the independent variable. volume of air, which is the dependent variable, and they have plotted it. Look at your intervals here, they're all equidistant apart, and the same goes that way. How much air is taken into the lungs in one breath during deep breathing? We have to obviously go to deep breathing. There's it there. It started at 3 liters. And if you take it, this one is halfway there. Look at the big one here. From here to there. 2 to 5. So it'll be 3 liters. You have to calculate that, right? 3 liters. How many breaths did a person take in the first 60 seconds? Remember, one line up, breathing in, out. So the first 6 seconds, that will be around there. You need to work it out from that area there. Did it say 6 seconds? 60 seconds, obviously. I was getting worried there. 60 seconds, that's up to this point here. Where we go there, up to that point there. So it's 1, 2, 3, 4, 5, 6. You're simply counting the breaths. Name 2 muscles in a human body which are actively involved in inhalation, normal inhalation. Obviously, that we've discussed in the last question. That will be the intercostal muscles and the diaphragm. The part of the human brain responsible for regulating the breathing rate. In other words, which part of your body, of the brain, controls the rate of breathing? And that would be the medulla oblongata. With that, time for you, Abby.
So much. You know, time is always an enemy. But otherwise, guys, learning continues. Bring all your questions because we're here to assist you guys. We'll see you after the break. Welcome back, Awesome Minds. It is now, since you've been hearing about the Safeways competition, if you think you are a bright spark, all you need to do is to jump on to facebook.com/SafewaysK53, caption the picture that we have there pinned on top, and the comment, the caption with the most likes gets you in a voucher worth 150 Rand. It's a Google voucher. So guys, if you don't have an app, you can buy the app and get some change to also buy other things. Competition is closing tomorrow. But now, thank you for the answers that we have on the challenge question. Aslam, it's time to answer the challenge question.
That's correct, sir. Are we ready for that?
Yes, yes.
Okay,
sir.
the first one, question 1, which we did not have much response for, am I right?
Yes, you
Albert?
are.
Explain how the function of the kidney will be affected if the ureter is blocked by a large kidney stone. First of all, the ureter's function is to take urine from the kidney to the bladder. So if it is blocked by a large stone, it's going to affect the flow of this urine from the kidney to the bladder. That's our first point that we have to make there. Let's elaborate on that. Urine will not be able to leave the renal pelvis of the kidney, causing swelling of the kidney tissue and accumulation of fluid in the renal tubules. Can lead to renal failure or kidney malfunction may result. It can also cause an infection in that area. It may also lead to pain and bleeding because the ureter is a narrow tube. If there's a stone, it's going to affect the walls of the ureter. So it's going to scrape against those walls. Those that have kidney stones will understand what I mean. I have several every now and then, so I know what I'm talking about here. Obviously, the first, for us, the main part is this one here, that the urine, I won't say leave the renal, but will not be able to move from the kidney to the bladder easily. It doesn't mean it'll get totally blocked there, but this one here said that it's a large kidney stone, so it's going to block it. And obviously that's going to cause swelling because the urine is not leaving there, and that's going to be painful, and it can lead to an infection. Question 2, and I believe we had some answer there. Yeah?
Yes.
We'll discuss the answer first, and then we'll comment on the learner's response. Explain how the body responds to dehydration on a very hot day when one is involved in strenuous exercise. So dehydration, losing too much of water. So in other words, the body has less water. That should already tell you, when we're talking about water, that means we are talking about osmoregulation. And notice I'm putting it here because after this we're going to do the kidney as well, so we'll talk about it in more detail. And therefore you need to know that the hormone involved is ADH, which is released by the pituitary gland. Now that's going to form the basis of our answers. So when the volume in the blood decreases, because we had to say that dehydration means that we've got too little water. Osmoreceptors in the hypothalamus, this is the control center for osmoregulation, are stimulated and messages are sent to the hypophysis or pituitary gland to release more. You have to say this word more because the hypophysis is releasing ADH from time to time. But now it's going to release more ADH. And the function of ADH is to make the walls more permeable to water. Of which walls? The distal convoluted tubules and the collecting tubules or collecting ducts. Makes them more permeable to water. What this simply means? Allows more water to pass through. This causes more water to leave the tubules via osmosis, or causes more water to be reabsorbed. And by the way, mindsetters, reabsorb means, notice the word, it's not absorbed, reabsorbed. Reabsorb means it was there, it went away, now we want it back. Where was it? It was in the blood. What happened? In the nephron. In the nephron. Filtration took place and it went down there. There was this water and it went in here. Now the water is in the tubule and we are short of water. So we want this water back. This is what we mean, reabsorb. From the blood it went to the tubules, and from the tubules we want it back to the blood. This part is called reabsorption. From the tubules back to the blood. Okay? Let's go back. The amount of water in the blood now increases back to normal, and more concentrated urine is formed. Concentrated urine means the urine has less water and more solutes, more salts. So that means the urine is going to then, so less water will be excreted by the body. In this way, we conserve water, we save water. And one of our learners was close to this answer, am I right?
Yes, you are.
And her name is?
Her name is Renali Teapo.
Good.
You know what that
Congratulations, young lady, you did very well. Let's go to our questions now. Okay, so while we're talking about the kidney, let's look at the question here. Study a diagram of a longitudinal section to a human kidney and answer the questions that follow. They're giving you several labels, a beautiful diagram given to you. Identify parts D, E, and F. D, look at the arrow. There's a blood vessel. Look at the arrow. The arrow is coming out of the kidney. So that will be the renal vein. E is this area here, will be the pelvis. And because it's in the kidney, we say renal pelvis. And F, where's F? F is going to this area here. This could be the medulla. And if it points to one of these guys, then it would be the pyramid of the medulla. Okay. In this case here, they're asking for the calyx, sorry. The calyx in that area, calyx in that area. This part here, they're pointing here. Look at this diagram, sorry. Let's look at this one here. It's pointing to this just below the pyramid of the medulla. They're showing you that area, so that's the renal calyx, okay? Why in an actual specimen is region B darker in color compared to region C? This area here, darker in color than C. That is because, and if you know your structure, you would know that there are more nephrons. B contains more glomerular capillaries than Malpighian bodies, or C contains no of that ones there. So we are saying that in this area there's more Malpighian corpuscles or glomeruli, so therefore it will be darker than C. Identify part A and state one function of it. What is part A? Okay, that is the renal capsule. Obviously, anything that covers something would be protection. Straightforward question. diagram. This is one way in which the kidney is tested. Diagram of the longitudinal section, macroscopic structure, and questions asked thereof. Another way is when they give you a diagram, of the nephron, and they ask you questions, or they don't give you the diagram, they ask you in the form of a graph. So they're telling you the concentration of certain substance as they move to various parts of the nephron. So here's the graph now. They're telling you this is part A, B, C, and D, or whatever. They're telling you it's distal convoluted tubule and the renal pyramid. So obviously, A is before all of that, and B is just after A. This A would be the Bowman's capsule, and this will be the proximal convoluted tubule and also parts of the loop of Henle. Name the regions indicated by A and B. There, I just labeled it for you. Bowman's capsule and proximal convoluted tubule. Which of the substances indicated on the graph is not excreted as part of the urine? So what are they talking about here? They're talking about water, sodium, glucose, urine. Is there water in the urine? Yes, otherwise the urine would be dry. We need some water in the urine. Is there sodium in the urine? It's a salt. Yes, there could be some urine, sodium there. Glucose, let's think about that. Urea, definitely, because urea is a nitrogenous waste, it has to be there. The substance that will not be there would be glucose. Because glucose is a useful substance, and we need all the glucose that we can keep. If we are excreting glucose, it means we have too much of glucose, and that means the person is diabetic, or they've had too much of glucose. Which product, according to the graph, occurs in higher concentration in the urine than in the blood entering the kidney? Now you've got to look at your graph. That's water. Obviously, it's coming down. Sodium, coming down. No. Glucose, coming down. Urea starts lower and becomes higher. So in the urine, that's here in this area. Once we're in the renal pyramid, remember, the renal pyramid goes to the calyx goes to the pelvis, ureter, ureter, bladder, bladder, urethra, out. So you're going to follow that pathway. So in that case, we are saying that it is urea, obviously. Explain the changes in glucose concentration during its movement through region B. So as you can see, first of all, even if you didn't know the story about glucose, look at the dotted line. That's the dotted line. In A, it was fairly stable, and the concentration of glucose in B, obviously, you can see what's happening there. It decreases. So that part you can see. Now, they wanted, but they didn't say describe what is happening. They used the word explain. Explain means you must say that it's decreasing, but explain why it is decreasing. I just told you just now, glucose is a useful substance, therefore it needs to be reabsorbed into the blood. Again, remember what I said about reabsorb earlier, from the tubule back to the blood. So these things have gone through to the tubule. Their destiny should have been urine. But if we need them, we take them back. And we don't allow them to go through. And that's what's happening to glucose here. So first of all, when entering, it is in a high concentration. We said that. When leaving, it is in a low concentration. Why? Because active reabsorption takes place against the concentration gradient. What does that mean? It is taken from a low concentration to a high concentration. Normally, things will diffuse from high to low. Now, here we're saying active. For active, that means there must be energy. We are using energy. It's like taking a rock and pushing it uphill. You need energy to push this rock uphill. Whereas if it was on the top of the hill, you just have to touch it, it will roll on its own down. That's down the concentration gradient. Now we are talking about against or up a concentration gradient. The transport mechanism requires energy supplied by the epithelial tissues. The mitochondria in the epithelial tissue will supply this energy. Some of the glucose is also used by the nephrons to supply energy. Because the nephron has cell, the tubules have cell, they need some of this glucose for energy, so they use some of the glucose as energy, and the rest can be reabsorbed by the... So in other words, we should have a zero amount of glucose when we come to the urine. That's what we are saying there. Explain now the mechanism that leads to a decrease in water concentration in the distal convoluted tubule. We're not going to discuss this answer because the question is different, but the answer is the same as AB, our challenge question.
Question, yeah.
That one talked about dehydration, what happens. So here we're also saying that if, what is happening here? We had lots of water in the tubules here. All of a sudden, we have less here. That means some of this water must have been reabsorbed or used up. And the mechanism, they're talking about the mechanism, so we are talking about reabsorption again. And it's the same story there. Osmoreceptors, blah, 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 the whole story. I'm not going to go through that again. We've done that. We've covered it. You should be au fait with that one now. Okay. Question 5, dealing with glucose levels now. They're telling you that the normal glucose level is 1 milligram per centimeter cubed, and normal insulin is 0.5 milligrams per centimeter cubed. And they've given you the whole story there, and they're telling you this is the story here. So glucose and insulin levels over time. The key blood glucose is the one with the triangles, the solid line, and insulin is the one with the dotted lines. Okay, here you have the concentration of both over time. What is the glucose level at 3 hours after the start of the investigation? So 3 hours, you're going to go there. We're going to the dotted line, we're going there, going across 1.4 would be the answer. So simple reading the information from the table, from the graph. Remember, people, you have to put the units because sometimes they give you a mark for the answer and 1 mark for the unit. So even if you get the answer wrong, at least you get 1 mark for the unit. 512, explain the relationship between insulin and blood glucose by referring to the graphs. So we talk about glucose because glucose is going up first. As the levels of glucose increase, what happens to the levels of insulin? It also increases. And that brings the glucose levels down, and therefore the insulin levels go down until the glucose level starts going up again. So that is what we are saying. An increase in glucose level results in an increase in the level of insulin. You cannot say an increase in insulin causes an increase in glucose. That's wrong in the first place in this particular case anyway, because when insulin rises, it converts glucose into glycogen and fat, and it stimulates the cells to absorb and use more glucose, and therefore the levels of glucose will decrease. This is known as homeostasis, and this is known as a negative feedback mechanism. Why? Because the response, the stimulus was lots of glucose. What was the response? Less glucose. So a negative feedback mechanism is that mechanism where the response is opposite to the stimulus to bring things back to normal, because this is homeostasis, maintaining a balance. You need to know this because this work here, especially this glucose story and the one on ADH people, is tested again in grade 12. You do it again next year under homeostasis. Name the organ in the body which produces insulin. That's fairly easy. It will be the pancreas. Explain the homeostatic mechanism that controls the glucose levels in the body. I explained it just now. So when abnormal levels of glucose are detected by the pancreas, in other words, abnormal, when the blood level of glucose rises, insulin is secreted to decrease the blood glucose level back to normal. Insulin secretion is then inhibited because the glucose is back normal. When the glucose level falls for some reason, for example, you're fasting or you haven't eaten for a long time, then pancreas secretes glucagon. Don't confuse glucagon with glycogen. This is a carbohydrate, and this one is a hormone. So glucagon is secreted to increase the blood glucose level back to normal, and the glucagon secretion is then inhibited. By the way, while we are talking about that, when we say insulin decreases sugar levels or glucose levels, what do we mean? We mean insulin converts extra glucose or sugar into glycogen and fat to be stored. Both these are insoluble, so they will not affect the concentration. At the same time, insulin stimulates the cells to absorb and use more glucose. All of this decreases that. And when we talk about glucagon, we say that glucagon increases the sugar levels. How? It converts this glycogen now to glucose. So it converts the glycogen that we stored into glucose. This will raise the glucose level. Good. The diagram below shows parts of the human digestive system and associated organs. Study the diagram and answer the questions that follow. There you got the diagram. Give the letter and name. Letter and name. Instruction is important. If you give either one only, you lose half the marks. Of the part where bile is temporarily stored, that should be B, the gallbladder. The hormones that control the blood sugar level are secreted. That means those hormones. It will be C, the pancreas. We just discussed that just now. Name any 2 types of enzymes that are found in D and briefly describe the role of each. D is the stomach, and the enzymes that are found there are carbohydrates, proteases, and lipases. These will break down carbs. These will break down proteins, and these will break down lipids or fat. List 2 functions of bile. That's simple, straightforward question. It emulsifies fat, it adds fluidity, alkalinity, etc. Those are the functions there. 3 functions of water in the movement of food. Easy. Acts as a lubricant, allows for peristalsis to take place, keeps the tissues moist, soft, and pliable, and for easy movement of food. Again, you note this question. It says food along the digestive system. We can also say that water is used for hydrolysis. In other words, to breaking down large molecules. Okay? Abby, you ready? Not yet. Okay. Name 2 substances that can be converted into other substances inside organ marked A. And that would be glucose, amino acids, and fat. And that's all we have for you guys today, and I'm hoping that we've helped you along the way. Notice there's still a little bit of population studies that you have to also do. Study that as well, so you are ready for this paper 1.
Thank you so much, Aslam. Until next time. Guys, thank you for tuning in. Remember, right now it's 10 o'clock. Check out the video that we have for you on the Safeways K53 page. But otherwise, thank you also to MTN SA Foundation for proudly sponsoring this show. We love you guys and all the best. Peace.